Thank you. Thank you, Bronwyn, and good evening, everyone. It looks like the next time we need to come up with a harder quiz. As you said, most people answered the questions correctly. Okay, let's start out with the river that we will be talking about, the Potomac. Uh, as most of you undoubtedly know, the Potomac starts somewhere here in West Virginia, and then it flows. It becomes the border between Pennsylvania and West Virginia, and then the border between West Virginia and Maryland, and then Maryland and Virginia, and then it empties into the Chesapeake. Now, my survey, the last 3 years, I've been surveying the snails in this section of the river that I marked with red arrows, starting at Violet's Lock, that's just to the right of the mouth of the Seneca Creek, and ending around slightly to the west of Brunswick. The distance between those red arrows along the river is almost exactly 50 kilometers or 31 miles. That may look short relative to the entire length of the river, but that's it's long enough for one person. Okay, now a little bit of basic biology. What is a snail? A snail is a gastropod mollusk with a shell. Here's a snail from the Potomac, Gillia altilis. It has a shell. Every turn of the shell is called a whorl. So, and the last turn is the body whorl because it sits on top of the snail's body. This part is the spire, and then at the apex we have the protoconch. The protoconch is the shell that the snail makes before it hatches out of its egg or before it is born. And these are the tentacles. That's the mouth. That slit you see in front of the snout is the mouth. I'm sorry, this is the snout, and the slit you see in front of the snout is the mouth. There's an eye. There's another one on the other side. And that's the foot, and there's an operculum. What is the operculum? The operculum is what closes the aperture when the snail pulls its body into the shell. That's called this opening is the aperture, and the operculum fits neatly into the aperture. All right. Here's start with a video of Gillia emerging from its shell, and there's the operculum. So it's that's the foot. It's searching for the ground under it, and it finds it. Now it's going to flip its shell. Now notice that the shell sits on top of the operculum when the snail is crawling. That's the foot. Okay, that's how it functions. All right, so what I said, the snails were mollusks. So what is a mollusk? Mollusks are members of the animal phylum Mollusca. There are several groups of mollusks. We have gastropods, snails, and slugs. Gastropods without shells are called slugs. There are marine and land slugs, but there are no freshwater slugs. We have bivalves, mussels, clams, scallops, and then there are cephalopods, octopuses, and squid, chitons. There are polyflacophorans, and then there are tusk shells, scupopods. These are all marine groups, and there are others that I'm not going to even mention. All right, what makes a mollusk a mollusk? Mollusks share several morphological characteristics, including the mantle and the radula. The mantle is this tissue you see here that covers the dorsal body of a snail or a mollusk. Okay, octopus has a mantle too. It's right here. What's below the mantle is the mantle cavity. Mantle cavity is important because that's where all the important organs are. All right, mantle cavity, and we have the operculum again. So, and the octopus's mantle is here, and its organs are inside. You can't see them, but this funnel here opens into the mantle cavity. Okay. All right, the second characteristics that is shared by most mollusks, except the bivalves, is the organ called radula. Radula is like a belt, a ribbon that has tiny teeth on it, and it sits inside the mollusk's mouth. That's the mouth right there. This is Gillia again. It's floating upside down on the surface of the water. This was a captive snail. It would be difficult to see this in the Potomac because this is not a very large snail. But at home, it was in a little dish, and it was easy to watch it and film it. Now, many freshwater snails do this. They float on top upside down. Now, this is a video clip of Gillia floating, and you will see its mouth opening and its radula coming out. It's at a half the original speed. Now, that thing you see is the radula right there. This is the radula, and these the rows, the white rows, are the teeth, the individual teeth that are on the radula. And so this is how they feed. They scrape off the food from a surface, or they pull in bits of food using the radula into their mouth. Okay. Now, here is the actual radula that I extracted from an animal. This is the radular ribbon. These are the rows of teeth. There are different types of teeth. This one is the central tooth right here. There are some, this is the main tooth. There are some smaller ones on the right. I had to squish this between a microscope slide and a cover glass. So these actually stick up, but in this picture they look flat. They're actually like this. These are the lateral teeth. They actually look like this. Okay, a little bit of taxonomy. We start with kingdom Animalia, and then we have phylum Mollusca, class Gastropoda, order Architanioglossa, family Viviparidae, genus Campeloma, and a species Campeloma decisum. Here it is. This is the basic hierarchy of taxonomy. Now, the zoological nomenclature follows the International Code of Zoological Nomenclature, a set of rather complex rules. But briefly, the complete name of an animal consists of a genus name, the first name, and then a species name. These are written in italics. And they're supposed to be Latin words or Latinized words from other languages. Usually Greek is used. What makes taxonomy confusing is that many species have been described by different people on different dates under different names. Usually they oldest name given to a species is the valid name of the species. And to lessen confusion, the name of the person who first published a name is usually given after the species name. So this snail was described by Say using this name, Limnia decisa. But now the species is in the genus Campeloma, and the name became decisum to agree with the rules of Latin grammar. And because the genus name has changed, we now place Say's name in parentheses. Okay, so you will be seeing both types of names during my talk. I'm mentioning this so that you won't be confused. All right. So snails are abundant in the Potomac River. How do you find them? Some of them live on or in mud. So the C&O Canal towpath is right behind these trees. And if you're taking a hike and if you see the river and then if you say to yourself, oh, hey, let me walk down the slope to the shore and look for some snails. And then when you get to the shore, you will look down at your feet and then you will say, hey, where is my right foot? The shore is usually like this. It's just very deep mud. There are snails here, but you can't really get very close to the shore. And there are islands. There are many islands in the Potomac, and their shores are good places to look for snails. There's even a Paradise Island. If you don't believe me, look it up on Google Maps. It's between Nolan's Island and Birdsall Island. So how do you get to where the snails are? My recommendation is that you get in your kayak and paddle out, especially if you have any doubts about anything. Here is an island, a little island, very flat, very low. It undoubtedly goes under the water when the Potomac starts to flood. The Maryland shore is on the left, Virginia is on the right. So if you want to get to a place like this, you need a kayak. All right. There are two evolutionary groups of freshwater snails. The first one is subclass Chinogastropoda. They obtain their oxygen from water using a gill. Shell aperture is closed with an operculum, and their sexes are separate. So there are males and females, but in rare cases, some species are parthenogenetic. That means there are only females, no males. Second group is subclass Heterobranchia. They obtain their oxygen from air. Even though they live in the water, they come up and they get their oxygen from the air using a lung. Sometimes what is known as a pseudogill may be present. They don't have an operculum, and each snail has both male and female organs. They're hermaphrodites. Okay. Shell types based on coiling. This is useful for preliminary identification. Shells of most marine, freshwater, and land snail species are dextral. That means when you hold a shell like this with its apex up, and this is the aperture will be on the right if it's dextral or right-handed. That means you can put your right hand around the body whorl. Some species are sinistral. They're left-handed. The aperture is on the left, and your hand goes around the left of the shell, your left hand. Some have very, very flat shells. They can be dextral or sinistral. It's usually difficult to tell just by looking at the shell. You have to look at the snail, the organs. The snail's organs are not symmetric within the body. Some organs are on the left, the others are on the right. So if you dissect the snail and look at the organs, then you can tell whether it's dextral or sinistral. And then we have limpets. They don't have any apparent coiling. They look like tiny cones. Okay, let's move on. Here we are, the group 1 snails. These are the snails that breathe in water. Here's Gillia altilis again. That's the mental cavity. That's the snail's body. And there's the gill inside the mental cavity. Those are the filaments of the gill. The water goes into the mental cavity, and the oxygen is extracted by the gill, and it goes into the blood. All right, 2 most abundant snail species in the Potomac River. Leptoxis carinata. If you are kayaking and if you go over rocks and in the summer, early in the summer, you're likely to see thousands of these little black things in the water or on sunken logs. Those are, most of them are going to be Leptoxis carinata. This is what the shell actually looks like. All right, that's the name of the snail. Leptos in Greek means thin, and carina is Latin, means a keel or carina. So this is the snail with a thin carina. The name refers to this edge you see around the shell. That's the carina, goes all the way around. These are juvenile shells. They're about 5 millimeters high, and these arrows point at their protoconchs. But look at the adult. It doesn't have a protoconch, it's missing. In adults, the protoconch always wears off. All right. Second most abundant species, Elimia virginica. It lives on mud. And when it's abundant, this is what the mud looks like. You see these tracks they leave behind them, thousands of them. Here's one, a single one. It's making its way through some sediment on top of a rock. And because they live in mud, their shells are always dirty, shells of live snails. This one here has algae growing on it. Here's another one as the operculum is trying to get out of its shell. They grow up to actually longer than about 30 millimeters, longer than an inch. But if you find empty shells, they're kind of pretty. They have, some of them have this dark colored band, and there are very fine lines and spirals on the shells. Now, they too are missing their protoconchs. You cannot, even the live ones, you can see they're all truncated, all the shells. The apex doesn't survive into adulthood. So the question may come to your mind, well, is there a hole at the top of the shell? No, there isn't, because the snail plugs it up even before the hole opens, even before the protoconch breaks off. All right. Campeloma decisum again. This is the largest native snail in the Potomac. It lives on mud or in mud. The shells usually have this greenish tint. There's the operculum. This is the largest live snail I have so far found, 41 millimeters in height. This is a species that has several interesting traits. All right. Normal shell coiling is dextral, but occasionally you may find a sinistral shell. Here are these are dextral, and here is a sinistral one that I found in the Potomac. Here is a live one, 2 dextrals, 1 sinistral. I have calculated that about 0.3% of Campeloma shells in the Potomac are sinistral. So if you collect about 300 of them, you may find 1 sinistral shell. Okay, Campeloma is the largest native snail in the Potomac. What is the largest snail? Cipangopaludina japonica. For some mysterious reason, these snails are called mystery snails. This is the largest live one I have so far found. Unfortunately, Cipangopaludina japonica and the closely related Cipangopaludina chinensis are not native to North America. They are native to Southeast Asia and Japan, as you can tell from their names. They're edible, so they were first introduced to North America in the late 1890s. And they're now all over the continent. This is a juvenile. It too has a slight keel around its body whorl and the upper whorls, and there are these fine, very fine spiral lines on it. Cipangopaludina also lives on mud, so does it compete with native snails? So far, there is no evidence that it does. I've seen on at least 2 occasions a live Cipangopaludina next to live Elimia at the same location. There's one and there's another pair. They're side by side. All right. These 2 species are in the family Viviparidae. As the name implies, they give birth to live baby snails. They don't lay eggs like most other snails do. Every now and then, if you're at the shore, you may find an empty or the shell of a dead Campeloma. And if you look inside, you may see the shells of unborn embryos still inside. So here is a Cipangopaludina japonica with a newly released baby snail. It's hard to tell that it's a snail, but it is, believe me. It has come out of the mantle cavity, and it's sort of slowly making its way down the mother's snout. Okay. Group 1 snails that I've been talking about, they have separate sexes. So how does one tell whether a snail is male or female? You've probably been wondering about this all your life. Now I'm going to answer it. In the family Viviparidae, the right tentacle of the male function as a penis. This is sometimes referred to as a verge. So here's a female. The 2 tentacles are more or less symmetric. They have equal lengths. But in the male, the right tentacle, this is the snail's right tentacle, is enlarged and lengthened. And that's what the male uses its right tentacle as a penis to inseminate the female. And here is a female from the Potomac. Both tentacles are the same. I have, using this method, I have sexed about 40 or 50 Campeloma from the Potomac, and they were all females. So my suspicion is that this species is parthenogenetic. So there are no males. Okay. The snails in other families have a separate penis. And so to tell the sex of a female, to tell the sex of a snail, you hold it with tweezers under your microscope in water. And when the snail comes out of its shell, you look at the neck. This is the neck of the snail. That's the head, that's the neck. If it's a male, you will see this appendage. That's the penis. The penis tend to be quite large, so you can't miss it. But the female doesn't have anything like that. So it's not too difficult. You just have to, it takes a little bit of practice. Okay. What about the smallest snail in the Potomac? It's Somatogyrus virginicus. It's next to a Cipangopaludina, grows to be about 2.5 millimeters. It has very long tentacles, as you can see here. Okay, now group 1 snails, they breathe in water using a gill. I have one more example, but that's an odd one. A long time ago, back in 1972, someone named William DeWitt published a paper about a snail called Pomatiopsis lapidaria. And he had found it near the Potomac River at a place called Fletcher's Boathouse. When I read this paper back in 2009, I said to myself, wow, I wonder if that place is still there. Well, it turns out Fletcher's Boathouse is still there. It's just slightly northwest of Georgetown in a place called Fletcher's Cove, and there's still a boathouse there. So I went there in 2009 and 2010, and lo and behold, found Pomatiopsis lapidaria. And my colleague Tim Pierce and I wrote a short paper about it. And you can see in this picture that these snails were not in water, they were on mud. They're group 1 snails, they have opercula. You can see their opercula right here. So what makes it odd? This is the anatomy of Pomatiopsis lapidaria. There's the gill. I colorized it to make it more obvious. So it has a gill, but it spends more time on land than in water. And some researchers actually think that it is a land snail. You're not likely to find it in the Potomac. If you're interested in finding it, look for it in muddy places along ponds near the Potomac, but not in the water. So next question is then how does Pomatiopsis breathe if it's not in the water, but if it has a gill? Here's a live snail. It's on top of a glass plate. It's out of the water. And if you shine light from under it, you're likely to see this air bubble in its mental cavity. That's the mental cavity under the shell. So after I noticed this, I thought about it for a while and then wrote a short note and put forward a hypothesis to explain the breathing of the snail. What I think is happening is that the snail still uses its gill to absorb oxygen from the water that's also in the mental cavity. But once the oxygen in the water is depleted, more oxygen diffuses out of the air bubble into the water, and then that too is taken up by the gill, and then eventually the air bubble runs out of oxygen, and when that happens, the snail just replaces the air bubble, just maybe pushes that out and gets in a new one. So that was my hypothesis published in the short note, and I don't know if anyone has given much thought to that process since then. Okay, now group 2 snails. Remember, these are the snails that breathe air. This is not an aquatic species. This is a land snail, but they tend to live near the water. I'm using this photograph because you can see its lung very clearly through its shell. Again, I'm shining light from the bottom. These are the blood vessels that are making up its lung. The lung is on top of the mental cavity. All right, Planorbella trivolvis, sometimes called the ram's horn snail. It has a very flat shell, about 15, 16 millimeters wide, and the shell is sinistral. And we know that from the positions of its organs. Its blood contains hemoglobin, and because of that, their bodies tend to have a reddish color. And they breathe air. Here's another video. Now, look at this. This is a breathing hole, and you will see it. This was, I filmed this in the river. The snail was right at the surface, holding on to some branch plant, the branch of a seaweed, and you will see the breathing hole open and then close. There, it's opening now. And it's closing. So that's how they breathe. They come to the surface and take in a gulp of air. All right. Planorbella trivolus is common in the Potomac, but it's rarely abundant. I usually find just one or two snails or one or two empty shells. This was one place where there was more than one in this little inlet. This was a rock island. The Potomac is right here. That's my kayak, my sunglasses, and you can actually see the snails. There's one there, there's another one there, there's another one there. And there was a crayfish right here, and that's right here in this video. And there's one of the snails. And let's see the crayfish move up. It's just, it's not doing anything. It's just going around. There's another Planorbella, and you will see some Elimia right here. So they all lived in this little shallow inlet. Okay, two more group 2 snails, Galba humilis and Physa acuta. This snail, again, they breathe air, and Galba is almost always outside the water, always on like mud and in wet places, and it rarely goes in the water. And Physa acuta, as you may have noticed, has a sinistral shell, and they don't have opercula. Here, you can find Physa in ponds and puddles. There are a lot of rock islands in the Potomac, and they usually have these depressions on them. Those depressions get filled with water from the river or from the rain, and there are usually Physa in them. So this was one such puddle. You can see there's one here, one there, and there was one there, and two others outside the water. Even though it was a dry and hot day, they were just sitting there outside the water as if they were land snails. All right, limpets. There are two. The limpets are in the air-breathing group, but their lungs are very small. Because of that, they have evolved this organ called a pseudobranch or a pseudogill. They use that to extract oxygen from the water. As far as I know, they don't come up to the surface like the other group 2 snails to get air. They just stay in the water. They live on rocks and small pebbles. They're not very large, about 4 millimeters. There are two genera in the Potomac. One is Physa, the other one is Laevapex. Their shells are slightly different, but difficult to tell apart. It's easier to separate them from their anatomies. Physa has clear, colorless tentacles, and the pseudobranch is not very obvious. On the other hand, Laevapex has tentacles with these black median lines in them, and its pseudobranch sticks out, and it has these laps, these folds on it. Okay, so here, this is Laevapex fuscus. There is the pseudobranch. You can see the folds on it. That's what it's using to extract oxygen from the water. And the tentacles, note these black center lines in the tentacles. And Physa does not have those lines, and its pseudobranch is less distinct. All right. Okay. Kayaking in the Potomac. Now, the best time of the year to look for snails is from late summer up to about early November. That's because the water will be low and the flow rate will be low. When the water is low, you can see the snails easier. But in the summer, it's going to be very hot. So if you intend to go out kayaking, make sure you have plenty of water with you, and your life jacket and your phone. If you intend to go kayaking in the winter, you're required to wear a life jacket. At other times, you're supposed to have a life jacket with you. Okay. The Potomac River floods at least once a year. And when it's flooding, don't go in the water. This is the point, this is the parking lot at Point of Rocks, boat ramp, last May, the 15th of May. That was the crest of the flood. And this is the same place when the Potomac is at its normal level. So don't attempt to go in the river when it's flooding. The flow rate is very high, and it's not very safe because there's always floating stuff, tree trunks, for example. You don't want to collide with them. The Geological Survey has a gauge at Point of Rocks. It's on the internet day by day, or actually it's continuously records the height of the river, flow rate, and other chemical various measurements, temperature, pH, so on. This was the height of the river during the May 15 flood. You can see it was about 25 feet or so. And look at the flow rate. This is the normal flow rate of the river. This was 2 orders of magnitude higher. You would stand no chance against a river that's flowing that fast. Okay. If you go up on the islands, it's fun to go up on the islands, but make sure your kayak is secured. Tie it around some rock or some tree. If you don't have anything to tie it to, clip it to your belt loop. You don't want the kayak to get carried away by the river. You don't want to get stranded on an island. And watch for tires. There are too many of them, unfortunately, too many of them in the river. Okay, I will end with some acknowledgements and thanks to the following: Maryland Department of Natural Resources for issuing me permits to survey the snails, Tim Pierce and Rob Dillon, who is here with us, for always offering help with identifications, and Natural History Society of Maryland, and especially Brownman, who is here with us, for making this presentation possible. And I will end with a bit of self-promotion. You may have noticed that of the 12 species I mentioned, 5 were described by someone named Say. Say was Thomas Say. He was an exceptional American naturalist, and he is the topic of my next lecture scheduled for March 19th. Details at the Society's webpage. Maybe Brown can tell us about it a little bit. Thank you for listening to me, and hope to see you all in March.
Thank you so much, Aydin. We'll go ahead and unshare. I'll put a spotlight on you, and we can open it up for Q&A. I knew this was going to be so interesting, and it did not disappoint. So thank you so much. You want to go ahead and hit unshare on your side?
Christina, how do I do that?
I can do it here. Okay, I got you.
Oh, okay.
All right, we got the spotlight on you. Remember, you can put your question in the chat box or raise your hand, and I will call on you. We do have a couple of questions already. Edith was asking, and we're not sure what species it was, but for the snail species whose shell usually curls to the right, but occasionally curls to the left, is there something different in its organ configuration? If not, how has it managed to survive?
Yes, when the shell is reversed, the organs are reversed also. How do they survive? That's an interesting question. That's something I'm actually working on right now. In Campeloma, as I mentioned, they are they don't lay eggs. So if people have been dissecting them, snails, and they a long time ago realized that I mentioned that the rate of reversal is about 0.3% in adults. But people a long time ago noticed that if you look at the embryos, there are more, the percentages of sinistral embryos are higher. It goes up to about 3 or 4%. So before they're born, there are more sinistrals, but after they're born, they don't survive as well as the dextrals, and they die out before they become adults. Why does that happen? We don't know. Something is wrong about them, just besides the reversal of the shell, of the coiling direction. Maybe something is wrong with their organs, maybe something is wrong physiologically, so they don't do as well as their dextral siblings, and they die out. But some do. I've shown you examples of live sinistral snails. They do. Some become adults, but not many of them don't. But I don't know the answer as to why they don't survive as well as the dextral ones.
All right.
Second question.
Yes. The question is there much difference between snail species above and below Little Falls with the tidal influence?
Yes, when if you get closer to the Chesapeake, you start to see some littorinids, other marine snails, but they don't come up to where I do my work because that's freshwater. But also in the Great Falls area, there is a non-native species, Virginia tentaculatum, I believe. It's been found there several times. I have not come across it. Apparently, it has not been able to cross into where I am surveying. I don't know the reason for that, but there is Cipangopaludina, the big one, the real big one. That's also non-native, but it's present there. But this the other one, the other non-native snail, is not present in that area. It's present where the falls are. It's also not very large snail. You may come across it. But where tidal influence, you will start to see marine species.
All right. The next question is, how can you tell the difference from the Japanese mystery snail and the Chinese mystery snail? And is one more common than the other?
That's a difficult one. maybe Dr. Dillon can answer it better than I can. I think the shells are slightly different. The, the Japanese mystery snail has more of a keel, I believe, but the Chinese mystery snail has a smoother shell. Is that correct, Rob?
Rob, go ahead and unmute. You're muted.
I sure am. You're exactly right, Aydin. The Japanese mystery snail, Cipangopaludina japonica, has a more angulate shell. It's got a stronger keel around the periphery, and the Chinese one is more smooth. And they really are hard to tell apart. If you hold them side by side, you can tell them. You can distinguish the two. But the primary difference is that there's the shape of the shell. The overall shape of the shell is slightly different. They're very similar. The Chinese one is much more common in the South. That's all we have here in South Carolina and North Carolina. The Japanese one, the japonica, begins to show up around Maryland, I would say, and then much further north, up in Massachusetts and Michigan and that kind of thing. Yeah.
Okay.
All right, we have a couple of people who are getting really jealous of you in your kayak. Do you have any favorite boat ramps or kayak put-in?
My favorite ramp is, they're all good except one. Which one is that? There's actually no ramp. You just have to sort of pull your kayak down these wooden steps. So that one isn't very fun. But all the others where there's actually a ramp, they're all fine. I like Point of Rocks. Brunswick is good. And then there's Nolan's is fine. You can use White's Ferry where White's Ferry used to be. It's not operating anymore, but there's a very large ramp there, and there's a restaurant right next to it. So if you get hungry on your way back, you go in there and grab a sandwich. So those are all fine. I've gone up Brunswick actually to there's a ramp at Dargan, and then if you go up the river, there's a place called Dargan Bend. It's a place where the river sort of suddenly turns. It's an interesting place. And then all the way up, there's Williamsport. There's a ramp also there, and there's Seneca Creek. There's a ramp on Seneca Creek, very close to the Potomac. There's a ramp on Monocacy, again, very close to the Potomac. You get on your kayak and you go under the aqueduct into the Potomac. So those are all fun.
Are there any species of snail that are endemic to the Potomac or any Maryland river that you know of?
Endemic? No, I don't think there are any endemics. No, I don't think so.
There's a little weird Fontagens bottomeri that it doesn't live in the Potomac. It's a spring dwelling or cave dwelling snail.
Oh, okay, right.
But you don't get your kayak into caves or any such thing.
No. No. Are there any other invasive snail species? I wouldn't really call them invasive. The Cipangopaludina, they don't seem to harm the local species. So I wouldn't call them invasive. There's, as I mentioned, there's another one, Bithynia tentaculatum. Is that the name? That's also non-native. It lives in the lower, around the falls, as I mentioned.
So Grace wants to know if there's any rare or threatened species, or are they all doing good?
About 45 years ago, a paper was published. And in that paper, I can't remember the name of the author now, and the author claimed that Elimia virginica, the species that I mentioned as one of the most common species in the Potomac, The author claimed that that species was actually endangered because he hadn't been able to find it in very many places. But now there are very abundant. Back then, perhaps the survey they did was very limited. And they were also, again, around that time, they were thinking that these non-native species were going to eliminate the natives, but that hasn't happened, and I don't think it's going to happen. So as far as I can tell, they are all seem to be doing okay.
And Alan is asking if you could do a little bit of a recap on what's the difference between the gill and the pseudobranch. And one snail has a gill, doesn't breathe in the water, but has an air bubble. And how does that air bubble get filled and refilled?
The air bubble, if you remember the photographs I showed you, the mental cavity opens to the outside. It's not sealed. So when Pomatiopsis is outside of water, when it's coming out of a shell, the mental cavity is exposed to the air. So air goes in, but there's also some water in there. So the air bubble gets trapped in the mental cavity. Right, so that's how the air bubble gets into the mental cavity. And I think when the snail pulls its body into the shell, it's sort of the mental cavity gets sort of squished and the air bubble probably just sort of goes out. And then the next time the snail comes out, air goes back in. What's the difference between a gill and pseudobranch? The difference is it's probably anatomical. A gill is always inside the mental cavity, but the pseudobranch is not. It's outside the mental cavity. It's, as you may remember the photographs, it's sort of on the side of the snail, that's sticking out, almost as if it's sticking out of the foot. So it's genetically probably, it doesn't come from the same sort of, during embryology, it doesn't come from the same, from the cells. It has a different origin. So it's anatomically, it's not a gill. A gill is always in the mental cavity. How many different types of snails do you see every time you go out to the river? It depends on how long how many hours I spend. Usually 4, 5, 6. Some of them, as I said, are common but not abundant. So you may not come across them all the time, even though they are there. And the small ones, the really small ones, are not easy to see. You have to really get out of your kayak and look around, look under the rocks, and those are difficult to see.
To follow up on Preston's question, what is the biodiversity? How many different types of species of snails have been found in the Potomac?
I have so far found about 13 or 14, but I think the actual number is slightly higher. If you look at the literature records, it may go up to maybe 15, 16, something like that. So it's about that much, about maybe 16. I haven't found, there are some species that I haven't come across yet.
Fred, you have your hand up. Go ahead, Fred.
Hi, good evening, Aydin. How are you? Wanted to ask you, you were talking about the dextral and central forms. It's interesting that you mentioned that the survival of the central is rather rare, 0.3, I think you said, percent, right?
Right.
With sea snails, marine shells, a similar rarity. As a matter of fact, among collectors, a left-handed specimen of any gastropod is always a highly acclaimed specimen and sought after by collectors.
Right.
And the one exception is the left-handed whelk in Florida, which is actually rather common. And I don't know if that anything like that exists in river snails, freshwater snails. There's a left-handed species. I've seen some marsh snails that are left-handed, and it seemed to be the common form. I mean, do you know anything more about that?
Right. Phycella are sinistral.
Okay.
Sinistral. Those small snails. They are sinistral. That's their normal.
Their normal morphology. Okay. Did you show that one? I lost track of. You
Yes,
had a lot.
there were
you
two,
did show it, right?
There were two species, Galba and Phycella. One was
okay.
sinistral, one was dextral, and then the Phycella was sinistral. Yeah.
Okay, I remember that. Yeah.
Phycella is their shells are always sinistral. Yeah. No, I don't survey bivalves. That requires a special permit because there are some endangered species. Actually, there may be some federally listed species, so that requires a different permit, and I don't have that, and I'm not going to try to get it. I'm just working with the snails.
And Alan, you have your hand up. Go ahead, Alan.
Hi, and Happy New Year. And you are the first to get this question this year that I ask, which is, can you remember your first interest in this area or what got you interested in this area in your life? And thanks again.
As Brahman mentioned, I'm actually a chemist by training, but I had always had an interest in natural history, especially animals. Several years ago, actually 1997, I remember the exact day, exact occasion very clearly. I was in Turkey. We had rented a yacht, and we were going around the coastline with some relatives, and we stopped in a little bay, and I decided to climb up get on the shore and climb up the hill just to sort of look around. And these were nice limestone hills, and the limestone hills are usually full of snails. So as I was going, I was not interested in snails at that point, but as I was going up this hill, I happened to see these little snail shells on the ground. And they were nothing like I had seen before. So I got very interested. I picked them up, and that's how it started. After I returned to the US, I looked them up and see what they were. And they just sort of, I went on from there. I started, picked up some books and did some reading, and got into collecting and studying land snails. And recently, about 3 years ago, I got interested in, because I was doing kayaking, I got interested in freshwater snails. I actually, my second or third time in the Potomac, I was on the Potomac River, and I was just kayaking, and I saw these snail shells, Elimia. They were Elimia virginica. I wasn't sure what they were. I took their pictures. I sent them to Dr. Dillon, and he said to me, "Oh, those are Elimia." Then I said to myself, "Well, why don't I start doing a survey if these are, if there are snails all over the place?" So that's how it started. Somebody was asking for my email. You can get it from Brahmin.
Yep, just you can email me if you want to get his email. I'll put mine in here. And Fred, you go ahead. You have another question.
Yeah, just a quick one on another note. You mentioned the Chinese snail had been introduced and possibly was for food, right?
Right.
Have you ever tried it?
No, I've never eaten a snail. No,
Okay.
they don't look appetizing. No.
No, they don't actually, but around the world, they're eaten in quite a few places. You might know. People eat freshwater snails quite a lot in many countries.
They eat land snails, freshwater snails. That reminds me, somebody was asking about invasive species. There is an invasive bivalve, corbicula.
Yeah,
The Potomac is full of it.
and they eat them.
They are also edible. I don't know if anybody eats them, but the Potomac is full of them. But I don't know if they are harming any native wildlife. They're
I've seen people collecting them by the 5 gallon bucket full.
oh, okay.
Yeah, they wade out into the shallows of the river and literally collect 5 gallon buckets full of these little corbiculas.
Yeah.
And
Yeah.
it's Asian folks because they know about them from where they come from. They eat them over there. So
They're very abundant. Yeah.
yeah,
I got two contributions on that. First of all, yes, you certainly can make chowder out of them, and in fact, they're corbiculin now. Second of all, goodness gracious, the Potomac River, are you out of your mind? Got, check the salmonella out there. Come on, man, get a clue. Stuff will kill you.
Well, speaking about eating them, what are the natural predators in the Potomac for these snails?
But what is the question again?
What are the predators of the snails in the Potomac?
I haven't seen any, but I'm assuming the crayfish probably eat them. I don't know about the birds. There are a lot of birds, but some of the snails, the ones that live in mud, for example, they would be difficult to get to by the predators because they are buried in the mud. But those that live on rocks, they may the crayfish may eat them. I don't know about the fish. Maybe they eat them too. Yeah, I don't know. I don't know the answer.
I'm pretty sure fish and birds eat them too.
Shorebirds of the fish. Okay. All right. Well, so there's so much for us to uncover here. Do we have any other questions for Aydin tonight? I did put his a link to his presentation coming up on Thomas Say in there. And Aydin did a presentation for us a few years ago on the slugs of Maryland, which you can probably still find on our YouTube channel, which is at Maryland Nature. So,
Browning, do you put these up on YouTube?
Yeah.
We could. We sure could. That's not a bad idea. Send a link to everybody there in the FWGNA group. That's a great idea. You did a great job, by the way, Aydin. And let me say this before we close. Your photography is outstanding. I mean, I've spent my career trying to take good pictures of freshwater snails. You must have some really good equipment.
Thank you.
It's just beautiful. I agree. I mean, to see the lung with the light coming through it, and to see the blood vessels in that, I mean, it was amazing, absolutely amazing.
Well, I'm glad you enjoyed it.
Yes,
thank you so much for doing this for us.
Yeah, thanks, Preston. Good to see you. Happy
New
everybody. Year.
Thank you. I'm glad you enjoyed it.
And for those of you with the left-handed and right-handed question, if you do look up Ned the Snail in New Zealand, as somebody found a left-handed garden snail, and then they had a whole nationwide hunt for another left-handed garden snail that could mate with them, because they got the story of Ned and they couldn't mate with a right-handed snail. And so it was a whole big thing in New Zealand earlier in 2025. But if you just Google Ned the snail New Zealand, you'll find out all about that. So that's just a side note.
Where do you find these things, Bronwyn?
It was on NPR. That was on NPR. But thank you so much, everybody, for joining us for our first Must-Learn Thursday of 2026. Look forward to seeing you next Thursday as we go through the micro world, more photography, and hopefully see it in March when Aydin comes back to talk about Thomas Say. In the meantime, stay well, stay curious, stay outside, and we're all going to be looking at snails and looking for snails, and you've definitely sparked an interest, I think, in a whole bunch of people for this slice of natural history. So thank you, Aydin.
Thank you very much, Aydin. Bye. Good night, everybody.
Bye.
Bye-bye.
Thank you. Thank you. Good night.